Welcome back. We've made our way back into the lungs now, so we're on our way through the conducting zones and into the respiratory zones of the lungs themselves. So we're now entering into the lungs. We came down through the trachea, and the trachea, as it gets to the last tracheal ring, that very last tracheal ring is a special one because it's kind of like a diverter, a splitter. And so we call it the carina. And so as we come down the trachea and get down to the very bottom, that last little portion that's there is going to be a special cartilage that splits off to the left and to the right, getting us into the left primary and the right primary bronchus. That's going to continue on into the lungs themselves. As we've come down through the nasal cavity, the pharynx, the larynx, the trachea, and we're making our way into the bronchi, all of the membranes that are there continue to warm, humidify, cleanse anything that happens to be in the air themselves, and that goes as well for the bronchi here. And so it's going to make sure that we get up to 100% humidity, so we're saturated with the water that's inside there, and we now have a fully humidified air coming into the lungs themselves. From the primary bronchus, it's going to then divide into a secondary bronchus, from the secondary bronchus into tertiary bronchi, and then from there we go into our bronchioles, and we're going to get smaller and smaller spaces as we move further and further into the lungs themselves. And so here's that carina, that last portion of the cartilage, and so it kind of looks like this in a sense for that last cartilage that's there. Mine's a little bit bigger in a sense than what it usually is, but just to kind of give you an idea of how it looks in a sense. And so it has the openings there. And then each of the tracheal cartilages, as they continue on up from there, move on up, and they're just the partial ring there, the 2/3 of the ring that we see on the rest of the cartilage there. So we then move into a primary bronchus that splits off into secondary bronchi. Those secondary bronchi then split into tertiary bronchi. And then we get into smaller bronchioles. And one of the things you'll notice in the picture here as well is that as we move from the primary bronchus to our secondary bronchus to our tertiary bronchus, we get less and less cartilage there. And so there's less of those cartilaginous rings there. They're starting to become more like plates rather than actual cartilaginous rings that are there as well. So we're going to get into these smaller structures that are there. So the cartilaginous support changes. And so we start to go from essentially circular rings to having just kind of like a plate here and there on the structures that are there. The epithelium is also going to change. We're going to start to move from a kind of protective layer into something that has the ability to move into gas exchange. So we're going to go from those pseudostratified columnar down into eventually simple squamous cells as we make our way in. As cartilage goes down, our smooth muscle goes up. And so that's going to be the overriding change that occurs as we move along. So we're now getting into our conducting zones here, and we're now into the bronchioles. The bronchioles themselves, we've made that transition from our pseudostratified ciliated columnar epithelium to now being in cuboidal epithelium. So we're kind of making that transition into thinner and thinner cells, so from a columnar cell down to a cuboidal cell that's there. We've got more smooth muscle, and we've got a large amount of mucus producing cells. So we have the ability to track more things in the fluid as we move into those bronchioles as well. So the respiratory zone begins at the point when we start to see alveoli. And so alveoli begins this process. And we can see this transition once we get to the terminal bronchioles. And so the terminal bronchioles start to feed into the respiratory bronchioles. And we're now into relatively small layers. We've got a larger amount of squamous cells in this area. We can start to see those transitions to being able to see gas exchange starting to occur. You have to remember when it comes to the lungs are not just a big bag in a sense. They're more like a sponge that's kind of sealed on the outside, so we can inflate them and deflate them as the air fills the spaces inside. But they have lots and lots and lots of small little spaces called alveolar sacs, which are just bundles of alveoli, so individual little small spaces inside the lungs. There are millions of these, over 300 million alveoli inside a healthy adult lung. And so we have just a huge amount of these structures inside the lungs themselves, and they make up the majority of the lung's actual total volume. And so all of those conducting airways, even a lot of our respiratory zones themselves, are not necessarily making up the bulk of the tissue. It's the smallest of the areas that's inside the lungs themselves, giving us just a huge amount of surface area for the gas exchange that we need to have happen. And this gas exchange happens very quickly. So here we have coming down through a terminal bronchus, or I'm sorry, through a terminal bronchial. As we move through that terminal bronchial, we come into a respiratory bronchial. That respiratory bronchial then splits into a different color here, splits into our alveolar ducts, and the alveolar ducts then feed into an alveolar sac. And inside that alveolar sac, we then have individual alveoli. So it's always been described as kind of something that looks similar to that of a bunch of grapes. And so if we have kind of like the larger stem here, and then individual bunches of grapes coming off of there, and then we have a cluster of a large number of grapes kind of on one little twig in a sense, and that's coming over the alveolar ducts themselves. We can even see these things when we look at them under the microscope. And so we can see the changes here. Terminal bronchial tends to have a little bit thicker of an epithelial layer there. That then feeds into a respiratory bronchial where it has thinned down to just about a cell layer thick, and then eventually coming down into alveolar ducts, and then those alveolar ducts feed into, here we have a nice alveolar sac there. Here we'd have another alveolar sac, and so on. And you can see that the terminal bronchioles, the respiratory bronchioles, the alveolar ducts, they don't make up a whole lot of the space. All of this space here is all alveoli. And so they're parts of other alveolar sacs. They're found within the tissue itself, and they make up the majority of the tissue that we see. Another example here, all of this space in a sense here is all alveoli, and just that small little portion there in the center here, this is where we have airways that are passing along. And so just a huge amount of space dedicated to the alveoli, and not a whole lot for, in a sense, kind of getting the air to and from those alveoli. Part of the job is the alveoli. The other part of the job is the blood supply. And so we have a barrier between our blood vessels, our capillaries, and that of the alveoli. And so in a sense, we're going to have an alveolar wall. We then have a membrane in between. So we have a basal lamina there, and then we have a capillary. And so for gases to move in and out of that particular space, they just have to go through a very, very thin wall, and that's it, in order for them to make their transition from oxygen coming in and CO2 going out. And so we have that change there between the two portions there. Inside an individual alveolus, so we have an alveolus here, we have more than one type of cell inside there. And so we have the majority of kind of the wall that is made up of that alveolus being made up by type 1 cells. And so we have these type 1 cells inside the alveolus. They make up the wall. These are the ones that are doing the gas exchange. Those are the ones that are allowing for that movement of gases in and out of the lungs and in and out of the capillaries. That is there. There are also some interspersed type 2 cells, and these cells produce surfactant. So they're going to be found in there as well, and they're going to allow us to help to make sure that when this alveolar wall is deflating, that they don't actually deflate all the way and end up sticking to one another on either side. We'll talk about that a little bit more in just a little bit. The other product that occurs, or the other thing that happens with our type 1 cells, is that they also assist in maintaining blood pressure. And so they're going to have the production of angiotensin converting enzyme. And so this is where, back when we were talking about the cardiovascular system and maintaining blood pressure, we talked about our renin-angiotensin system. And in order to convert our angiotensin into the active form, we need angiotensin converting enzyme, and that's going to come from the lungs themselves. So inside the alveoli, we have these type 1 cells, and that surrounds the alveolus itself. Outside of that, we then have a layer of elastic fibers that are going to help with the kind of the resiliency of the lungs themselves. They're going to allow them to inflate and deflate and not become damaged in that process. In addition, between adjacent alveoli, and let me erase that one and start over again. So between adjacent alveoli, we have these little pores. And can't really draw one there anymore, but there's a pore there as well. And that's going to allow the air that's coming into them to equalize in terms of pressure throughout the entire lung. And so the entire lung is basically going to have the exact same pressure all throughout, because all the alveoli are going to have the same pressures as well. In addition to those type 1 cells and type 2 cells, we also have macrophages. And these macrophages are, in a sense, kind of wandering around, making sure that anything that is potentially inside the lungs gets gobbled up and destroyed, in a sense. And so they get destroyed before they can cause us any kind of problems inside the lungs themselves. So those are resident macrophages that are inside the alveoli themselves. These are in addition to our type 2 cells, which are producing our surfactant, and then all of the walls are our type 1 cells. And so all of those individual cells there that are in between the different spaces there, those are our type 1 cells, allowing for gas exchange and the production of angiotensin converting enzyme. Different picture, but shows the same concept inside there. Here we can see one of those pores, so that's allowing that gas to maintain the same pressure throughout the entire lungs themselves. We've got our surfactant producing type 2 cell that is there. We've got our macrophage, and then we've got our wall of the alveolus, our type 1 cell as well. So when it comes to the lungs themselves, we kind of went into the lungs and didn't really look at the actual lung itself. We just looked at the airways that were there, starting with the primary bronchus and then working our way all the way down to the alveolus. Now we're going to look at the individual lungs themselves. And so we have 2 lungs, one on either side of the thoracic cavity. They take up the majority of the space there, except for the mediastinum, so where the heart is and all of our big vessels and our trachea and our esophagus and things like that are found within that mediastinum as well. All the rest of it is taken up by the lungs. And so when we have our lungs, the lungs have a base to them. They then go up and they have an apex. So the apex at the top, the base at the bottom on those. They have a couple of different areas that are there as well. And so in a sense, in the medial side of the lungs, I'm not drawing these to scale per se. We have all of the openings for our bronchi, our artery, and our vein exiting the lungs themselves, and that's going to be at the root of the lung itself there. The outer edge, in a sense, that is going to be touching the actual lungs themselves is the costal surface. So there's a lateral portion there. That's pretty much where I drew the line. There's going to be an anterior portion and a posterior portion as well. So that's going to essentially be going along the rib cage itself that's there. And where this root of the lung is as well, we have kind of an indentation. Go back to this color. And I can't really represent it all that great. It'll be on the picture here in just a moment. We have those structures there as well of the hilus, or the hilum. In addition to the lungs that are there, we have our left and our right lung. They are not equal in terms of their appearance, and so the left lung and the right lung are not completely shaped the same. The left lung is slightly smaller. It does have only 2 lobes, and so it has an upper lobe and a lower lobe within each one. And then they're separated by an oblique fissure that kind of comes across at an angle across the lungs themselves. The right lung, in addition to having an upper and a lower lobe, it also has a middle lobe. And so those 3 lobes are then separated by an oblique fissure again. And then we also have a horizontal fissure. And so those kind of come across there as well. Because the 2 lungs have I'm going to draw it even bigger there. They have to make room for our heart. And the left lung there has an impression for the cardiac notch. And so there's this space right in here is the cardiac notch as well. It's in there. And so here we can see that cardiac notch on that left lung, the little indentation that's there. It also has this whole entire space where the lung basically protrudes into it as well. We can see the oblique fissure on the left lung, and then our oblique fissure on the right lung, and then we also have a horizontal fissure as well. So we end up with a superior lobe on both sides. We have an inferior lobe on both sides, but then the right lung here ends up with an extra middle lobe. So because of that space that's there in between there. It also is larger simply because we don't have to make room for the heart. And so we have a little bit of extra space there that it can function on. The lungs receive blood supply from 2 different sources. And so we have blood that is going to actually perfuse the lungs and give the lungs their nutrients. And then we also have blood supply that's going to be coming in from the heart that's going to require the transition of oxygen and carbon dioxide in and out of the lungs themselves. So they really have 2 different sets of arterial circulations coming in. We have the pulmonary arteries, and then we also have coming in on the bronchial side. And so the pulmonary arteries are those that are coming from the rest of the body in a sense. So this is systemic venous blood. This is where we're going to essentially follow the bronchi in, and this is going to allow us to pull oxygen into the capillaries and move carbon dioxide out. And so this goes into those same spaces. And then they drain via the pulmonary veins. And so this gets us back to the heart. This is going to come back to the left side of the heart. So this is going to end up in the left atrium. And we now have oxygenated blood that can go out to the rest of the body itself. But the lungs need their own blood supply. So just like the heart doesn't take its blood supply from or its nutrient supply from that which is inside it, the lungs don't do the same thing either in terms of taking it from those pulmonary arteries. Not to mention the fact that the pulmonary arteries are also relatively deoxygenated blood. So they have their own bronchial arteries here. Those bronchial arteries themselves are coming off of the aorta, and so they're branches of the aorta itself, and they're going to basically follow the same pathway as the pulmonary arteries that we have, and they're going to make their way into the lungs and divide and divide and divide, and they're actually supplying all the lung tissue with nutrients that they need, except for the alveoli. And so the only place where, in a sense, it's drawing that supply from is from the capillary beds of the alveoli. They need to draw it from the blood that is flowing through those capillaries themselves. Just like the pulmonary arteries drain out through the pulmonary veins, we mix in this blood into the pulmonary veins as well. And so they're going to bring most of the blood back to the heart through the pulmonary veins. So we're going to have a little bit of a decrease in oxygen because we have a little bit less fully oxygenated blood coming back directly to the heart from the lungs because it's mixed there inside the pulmonary veins. The lungs themselves are surrounded, just like the rest of our organs, with a connective tissue covering called in this particular case, we call it a pleura. So we had the same thing over in the cardiac area there where we had our epicardium surrounding and their pericardium surrounding the heart. Here we have a parietal pleura. And so we have a parietal portion that covers the walls, and then we have a visceral portion that actually covers the lungs themselves. And so the parietal portion is the part that's actually against the chest walls themselves. This is a little membrane that we can see right inside the chest wall itself, and that provides for essentially kind of a seal inside the thorax. We then have the visceral one, which actually covers over the outsides of the lungs. In between the two, tiny bit of fluid. This is going to decrease the amount of friction as those lungs inflate and deflate inside the chest and as the heart's pumping and so on. So we have a little bit of pleural fluid that's there in between and keeps the two from rubbing against one another and creating too much friction inside there. That'll do it for this time. Next time we'll talk about the movement of the air in and out of the lungs and keep on going from there.